All right, so thanks for having me. This is my first foray into crayfish work just in the last couple of years. My PhD stuff was on freshwater snails, and then I started work on freshwater mussels, and then I started doing work on environmental DNA, where we go out and take water samples and figure out what organisms are present in the environment. And this particular project sort of kind of fell in my lap, which I'll talk about here. And after coming from the eDNA stuff, let me tell you, this stuff is sort of like a breath of fresh air. This is sort of what my training has been on. So it's been really cool to see some of the patterns that are in this data. So just some acknowledgements. Zach Couch is the one that sort of roped me into this. Günther Schuster has been a great source of information, and I have stolen liberally from his photographs for this presentation. I've gotten some laboratory assistance from undergrads, Tanya Safranek, Holly Martin, and then Robert Jackson when he was a master's student and after he graduated a little bit. This work was funded by Kentucky Fish and Wildlife Resources, and Tanya Safranek was supported by a Battelle Scholarship when she worked on this. So I'm going to jump right into sort of the project that got this project going, and that is Couch and Schuster's work in 2011. This was Zach Couch's master's thesis work when he was working under Dr. Schuster. He was doing a survey for this Orconectes jeffersoni, which is now in the genus Faxonius. So it's Faxonius jeffersoni. This is the Louisville crayfish. This blue dot's a little bit too big to show where it actually occurs, but it essentially occurs in Louisville. And in there, Zach noticed some of the populations look a little bit different. So he commented in the discussion, we should do some genetic analysis to see if some of these other populations are actually different species. So I'm going to play this little video. I think my audio is going to work. Basically, this has got some news press, and so you can hear directly from Zach on this project, since this is his. This portion is his, and this is his discovery. So you might also notice EKU graduate student Michaela Rogers in here, one of Dr. Dodd's students.
Biologist Zach Couch starts the morning looking for something he's already discovered, a new species of crayfish. At Fern Creek in Louisville, he and a fellow biologist with the Kentucky Department of Fish and Wildlife Resources look for them the old-fashioned way.
No go right there. Let's try one spot over here. Crayfish like to live under rocks, so you try to find the biggest rocks you can find and flip them over. And then kind of kick around, same way as we all did as a kid, looking for crayfish. It's the same technique we use now.
The journey to confirm Kentucky's 65th new species of crayfish took almost a decade. Couch made the discovery in the mid-2000s doing graduate research in a nearby creek on the Louisville crayfish.
Okay, this species, even though from a distance looks the same, when you actually get it underneath the scope and look at it, okay, this is something different.
But lack of funds after his thesis wrapped put the project in limbo until funding from his employer, Kentucky Fish and Wildlife, allowed Couch and a team to pick it back up, and the new species was confirmed early 2019.
I've worked with a professor from Eastern Kentucky University who's run all the genetics tests for us and provided the results to show that, yes, genetically this species is a new one and basically is independent from the species that we have over in Beargrass Creek.
Thankfully, finding the crayfish under a rock
here we go.
Doesn't take as long as finding out it's a new species.
Oh, there it is.
It's only found natively in Jefferson and Bullitt counties. One way to differentiate the new species is its claw.
So everybody, if you're a kid that grew up in Kentucky or in the Southeast and you played in a creek, you pulled out a crawdad that has that orange tip and a black band behind it. Well, what differentiates this species from the one in Beargrass Creek is that the thickness of that black band that you see is probably twice as thick on this species as it is in Beargrass Creek. So from a color standpoint, that's the easy way to differentiate between these two.
The last piece of the puzzle, naming it. But Couch isn't doing that. The opportunity goes to the highest bidder from Kentucky Wild's online auction. All funds go to support non-game species in Kentucky, like the crayfish.
The fact that we can discover a new species of crayfish right here in a creek that's between two apartment buildings shows you how much work we have left to do in the state.
In Louisville, Eileen Street, Spectrum News 1.
All right, so you heard her mention the genetic stuff. That's where I came in. Basically, he gave me 4 specimens, 2 from each, and I was able to wiggle them in on another project. And the preliminary results from that is what led us to a small little grant from Kentucky Fish and Wildlife Resources to expand the sampling. So total, we did 61 specimens from those 2 counties mentioned, Louisville and the surrounding areas. I extracted using a spin column kit, and I amplified a 600 base pair portion of the CO1 gene, cytochrome oxidase gene. This is traditionally the traditional DNA barcoding mitochondrial gene used for animals. And we did the Sanger sequencing off to Eurofins. I constructed a maximum likelihood tree, and I also generated a phylogenetic network using a program called splits trees. So I want to give a little bit other background. So this particular paper from 2006 by Taylor and Knopf, basically the work that I'm going to talk about today would not have been possible had they not undertaken this particular project. Much of the data that I'm going to talk about today comes from this particular paper. So basically, Taylor and Knopf were looking at gonopods. This is the male genitalia of crayfish, and it is one of the primary taxonomic identification features. So historically, many of these crayfish have been grouped into subgenera based on the shape of genital morphology. And the reason why people who study crayfish use this, and this is actually true for virtually all arthropods and a lot of invertebrates, that basically the genitalia is an indication of reproductive isolation, or that's what's sort of traditionally thought, that there's sort of the lock and key, and the lock is the female anatomy and the key is the male anatomy, and if they don't fit, then they are reproductively isolated. So in many invertebrate groups, and it's much easier to describe the key than is the inside of a lock. So in many invertebrate groups, male genitalia are used to diagnose species. And in crayfish, the overall shape of these has also been used historically in classification of subgenera. So the idea that more similar genitalia, they would be more closely related. Well, Taylor and Knopf did this really large sampling, lots of species from across the country. There's very little replication in here, but they did sort of a great job in getting lots of species represented in here. And these different colors are different subgenera, which are somewhat related to the morphologies you see on the left. And you can see just the colors are all over the place. So what this is basically saying that the mitochondrial genes do not reflect the gonopod morphology and potentially the historical subgenera that we really don't even use anymore. So this is not to say that the male genitalia are not important in terms of a taxonomic character. They're still useful for identifying crayfish, but they don't necessarily reflect the evolutionary history of the group. There's something else going on that's sort of driving the evolutionary history of the group, not the gonopods themselves. So just a note, again, that most of these things have been changed. The genus Orconectes is now only reserved for cave dwelling species, where everything else in this tree has been changed to, almost everything on this tree has been changed to the genus Faxonius. But some of the older papers that I'll reference here will have the genus Orconectes. So another sort of unique issue in sort of arthropods in general is the presence of pseudogenes. So since these papers have been published, every person who does a crayfish DNA paper has to say, I've checked for the presence of pseudogenes in these. So it seems like the mitochondrial genome has gotten dumped into the nuclear genome in many arthropod groups. Crayfish, other decapods, grasshoppers, it's been found in multiple places. And so those pseudogenes seem to mutate because they're not under a process of selection, they freely mutate. And when you go to try and amplify the actual mitochondrial gene, you sometimes get these pseudogenes. And the way that you can tell is that they actually have stop codons in them. So you can tell that when you translate them into an amino acid, they don't actually turn into a protein. Jen Buhe was actually a EKU graduate from, I think, the early 90s, something like that. One of Gary Richardson's students. And so I have checked for that. None of this is present in this data set, and it's sort of just a box that we now have to check off in the crayfish DNA stuff. So simply, I was brought on to say, are these genetically different? Are the two populations or these different river drainages in Louisville, are they genetically different? And so, like I said, I got basically 4 specimens, 2 of each at first, and then that led to the bigger project. So are they genetically different from each other? Well, I threw them in a tree, and at first I didn't know anything about these things, and I still kind of don't know anything about them. But I threw them in a tree, and I could say, well, here's one, here's the Louisville crayfish, here's the other. So they're different. You can see there are all types of other species mixed in here. They are different. So yes, they are genetically different. They differ by 4.11 sequence divergence when you do a pairwise comparison. So they are really different from one another. So the taxonomy portion you heard in the video, Kentucky Fish and Wildlife Service has decided to auction off the rights to name it. So they're looking for sponsorship. The money goes into Special Species Conservation Fund. This is their deal. Like I said, this is Zach's discovery. This is not mine. So they do have a sponsor. They have a name, but I'm not going to reveal it today. That's not mine to reveal. This is, again, this is Zach's deal. So I have just referred to this as Faxonius SPA, Faxonius new SPA. So I don't want to reveal it. Hopefully I didn't miss it on any of the figures that I have here as for preparing the manuscript. So that's their deal. I'm encouraging them, and it actually worked out really great. So I'm happy the way it turned out. So we know, yes, they're genetically different, and they're really different. But the thing that got me interested is like, why? These things are so geographically close. Why are they so different? So I'm a master at map making, which you can see here. So this is freeware GIS, Diva GIS, which is my go-to quick, low memory map making software. So in red dots there we have Faxonius jeffersoni, the Louisville crayfish, and in triangles down there we have the new species. And so I plotted this on the map, and you know, something popped out to me. So Faxonius jeffersoni, these all drain directly into the Ohio River. So this big river here is the Ohio River. And so right there is the falls of the Ohio River. And so that got me thinking, well, maybe that is an important feature that's sort of separating these things. But it was kind of unusual to me because after 15 years of studying phylogeography of fishes and mussels and things like that, nowhere has anybody ever talked about the Falls of the Ohio being a barrier to anything. It seems like organisms can go back and forth. And I'm not even saying in this presentation that it is a barrier. It might not actually be the barrier, but this was sort of the geologic feature that caught my attention. So this is a map of the Falls of the Ohio River. So you can see these series of islands at low water. Basically, this is historically went a drop of like 26 feet in a mile. So this is, there are some photos of what it would have looked like, portions of it anyway. But this is what it looks like today. So it's been highly modified. So in 1830, the Alpine Lock and Dam was completed, and that's here on the left side. So you can see basically they dug this out because the Falls of the Ohio was a barrier for shipping, for navigation from south to north. And this is essentially the only blockade through basically from New Orleans to almost to Pittsburgh, essentially. And so the entire town of Louisville was basically built up around to support getting around the Falls of the Ohio River. The city of Louisville likely would not have developed into the metropolitan area it is today without the presence of the Falls of the Ohio River. Later on, they also built a hydroelectric dam, which consists of 2 dams. There's a dam here, and then pretty much all the way across the island here is dammed up as well. So you can see the remains of the Falls of the Ohio, it's now a state park in Indiana, has been sort of walled off, and the rest of it has been flooded. So what sort of makes the Falls of the Ohio important? It was historically a migratory path for buffalo. So as bison migrated from the plains to the licks of Kentucky, so your big bone lick and your salt lick and all the other mineral licks of Kentucky, this would have been the shallowest part, and they would have likely crossed here. And there are buffalo highways in Indiana that lead to this area. And so this area has also been historically important for plants that have been dispersed by the bison, like the running buffalo clover, which has been studied by Dr. Koslow and David Brown, Short's goldenrod, which has been studied by Dr. Callie, and then this now extinct Falls of the Ohio scurf pea. It was only known from Rock Island here, and it went extinct likely about the time that the bison was basically extirpated from the area. So these things disappeared before the construction of the canals and dams and things like that. It was really the elimination of the buffalo that led to the extinction of this particular species. Fortunately, the running buffalo clover and Short's goldenrod, they are listed as endangered, but they have been found in other locations. But again, this really hasn't been a barrier for species. The only species that this has been a barrier for is us and our navigation and moving things via commerce. This is just a general map here, just in case you're unfamiliar with the area. We have the upper Ohio Valley and the lower Ohio Valley. Portions of the this was glaciated, which I'm going to talk about. I've been playing around with Adobe Illustrator, and for graduate students listening, if you don't know, your advisor can get you access to the entire Adobe Creative Suite. They can get licensed to install it up to 3 machines. So if you want Photoshop, you want Illustrator, you name it, you can get access through IT. Tell your advisor to call up IT and get access, and they can install it on a computer that you have access to. So this is the maximum extent of the glaciers. So notice the furthest south, right in Louisville, right here, would have been the maximum extent of the glaciers. So pre-Illinoisan, 2 million to 420 years, 420,000 years before present. The successive glaciations didn't reach quite as far south. So before the glaciers, the rivers were much different than what we see today. This is what were inferred to be the pre-Pleistocene river drainages. Many of these would have probably taken shape during the Miocene, when there's been some uplift of the Appalachian Mountains. These are the names of them. And in particular, I want you to note this Old Kentucky River. Note how it turns north here. It does not go into the Ohio River here. The Old Kentucky and Old Licking were not tributaries of the Ohio. They instead went into this Taze River system. And so this Taze River system would have been this sort of huge river system that drained much further north. And there are remnants of this. This is the Taze Valley in West Virginia. This is basically an abandoned river valley. So this river was essentially buried by the glaciers. You can actually go in places and they do road cuts, and you can find river sediments that have been buried from this massive river drainage. And so note, we're right there. Louisville, essentially, we're even smaller there with our 2 little crayfish, right in this area, which would have been the maximum extent of the glaciation. So again, we had the Falls of the Ohio there, and the Falls of the Ohio was basically carved from glacial meltwater. So as the glaciers melted, they scoured out what is now the present day falls. They also carved out this Harrods Creek here. This would have also been a glacial outwash area. So this creek wouldn't have actually been there until much later. So the creek that Faxonius jeffersonii hadn't mainly inhabit wouldn't have actually been there. So this is a glacial meltwater escape. The other interesting feature in this glacial history is the proglacial lake tite, right here over eastern Kentucky, West Virginia, and Ohio. Now this would have been formed from an ice dam, essentially, and this was a huge lake. And this particular study actually claiming they find sediments from this proglacial, this glacial lake tite in the Gulf of Mexico. You can actually find the alluvium from this lake. So eventually this ice dam would have broken and this lake would have drained. Now, similar to the way Lake Missoula and the scablands were carved out west, but this drainage would have taken 100,000 years. This was not sort of an initial really fast, it's all gone. This would have taken a long time. This lake was massive. There are competing hypotheses about the direction of the Taze River. So this particular map here, this would be the old Kentucky River, and the leading into the Taze actually think that it might have actually drained north into the St. Lawrence. And here is that glacial lake tight. So there are debates. I think the biology supports some of these more than others. The other thing to keep in mind is all these alternative hypotheses, they might all be true. They might be true in different time periods because these drainages have been moved around by the glaciers, so all these different hypotheses might be true at different time frames. There are also additional competing hypotheses about drainage divides. So this one, the Madison Divide in Louisville. So basically the Ohio River would have been just sort of a sliver of its current self, basically starting there, where the Kentucky River going this way up into the Taze. Other people think the divide would have been up here and the Kentucky River would have actually been connected to the Ohio River. Again, some of these might actually be true. They might all be true at different time periods. But can the crayfish DNA offer us some clues? So I made a big phylogeny. Here's my obligatory tree that you can't read. This is a phylogeny of the Cambaridae, the entire family that is found here in Eastern North America, and this is the genus Faxonius. I also took this data and put it into a phylogenetic network called splits tree. And this is what it looks like. So basically, instead of, this is almost like an unrooted phylogeny. And so the species are represented at the tips. And I was sort of blown away by this figure. I know it looks like a big tuft, but that tuft right there, it looks like there's some sort of shared history among this group. And so that tuft right there is represented by this little star on the tree. So you can see there's something different going on with this big tuft. And this is some representatives from them. So many of you saw Günther's color crayfish talk. This group is decidedly more boring than the crayfish featured in there. But you can see some commonalities in them. But in terms of crayfish color, these are the less spectacular ones. So this is the phylogeny for that tuft. And so there are species, so they are different, but do these broader species relationships reflect geologic patterns? And there is a lot in here and in that tuft that I'm not going to be able to talk about. So I'm going to just talk about these two particular clades. So first up, this clade here with Jeffersoni. So this clade has two, this has the Jeffersoni, the Louisville crayfish that we mentioned here in red, its distribution right there in Louisville. This clade also has Faxonius sloani, Sloan's crayfish, which is also sort of right on the margin of the glacial limit. And so these two species are actually nested within another species, Faxonius juvenilis. This is the Kentucky crayfish. If you leave EKU and go to a stream, the first stream that you find, you're going to find Faxonius juvenilis. So they are sort of nested within this more widespread species. And this is a pattern that is consistent with parapatric speciation. When you have this widespread species and on the margin of the range, you sort of have speciation just on the edge. And I think that's what's going on here. And so the result of that is paraphyly. So basically you're having these species nested within this more broad, widespread species. And interestingly, Faxonius jeffersoni is most closely related to juvenilis from the Kentucky River right here, Madison County. That is the most geographically proximate sample I have in this data set. And so there are other juvenilis samples scattered throughout here. They're geographically much further away. So juvenilis is much more widespread. It is here in the Kentucky River. It's also found in the Salt River and the Upper Cumberland and some glaciated portions of Southeast Indiana. Now, I want to make clear when I draw these maps, I don't think these crayfish were right up against the glacier margins. Their ranges would have sort of been ebbing and flowing. We don't have crayfish up near glaciers nowadays. They would not be able to tolerate these temperatures. So this is just really for illustrative purposes. But this distribution is kind of unusual. It's pretty disjunct. Note you have some drainages down here and then the Kentucky River and the Salt River. So this distribution is weird. And then nested within that is this other species, Durellii. This is the This is found in the Highland Rim. This distribution is a little misleading. It makes sort of a donut shape around the Nashville Dome, Nashville Basin here. But notice it's paraphyletic. So this juvenilis clade is more closely related to some, and then this one is more basal to it. So this again is sort of unusual. So I think a lot of these can be explained through patterns of stream piracy. I was calling this stream capture, and then I was giving the talk to Blair last night, or just running through it, and I one time said piracy. She's like, ooh, say piracy. So basically when a drainage is subsumed by another drainage. So a creek goes into another drainage. And we find evidence for these stream captures, not just in geologic evidence, but we also see it in the distribution of aquatic organisms. So this is the Kentucky arrow darter, a species that Dr. Harrell has studied. It has a disjunct distribution. I believe it's probably been split up or subspecies, things like that. Another one that's in this area, the orange throat darter, I believe. This one, note by Branley Branson and Don Batch, two former EKU professors. I like how in the title they have a footnote for the funding from an EKU institutional grant. Right in the title, they cite the funding source for EKU. So I'm not going to talk about all these, but I'm going to show a couple examples. Here is Girdler, Kentucky. This is south of Manchester. You can see sort of the relative placement of it. These streams up top here, this is the Collins Fork, and this drains into the Kentucky River drainage. Down here at the bottom, these are streams, Richland Creek, Little Richland Creek. This drains into the Cumberland drainage. And so there are some unusual fish distributions in this area. So if you look at a satellite image of this, you note this town of Girdler, which I note here is interesting. The Wikipedia page brags that they have a Dollar General and multiple churches. But the folks in Girdler have no idea, most of them I bet have no idea, they're actually living in the midst of a paleo drainage right here. So historically, the Collins Fork actually went down through here and was connected to the Cumberland River drainage. So basically the Kentucky River stole this river drainage, and it shifted course as parts of the Cumberland Plateau is eroded away, and this now continues up north through Manchester. So this would have been the route. So this explains one of the drainage jumps from the Cumberland drainage into the Kentucky. There's another example in the Salt River. I did not make this map, apologize for the contrast, but this is Lawrenceburg up here, and this river here is the Salt River, and note how it makes a sharp west turn. Well, this was also a stream capture. This used to go to the Kentucky River. So there is also a paleo channel here. This might be better illustrated by this diagram in the bottom left. This in black here actually was all part of the Kentucky River. And there's been a lot of back and forth exchange between the Kentucky and the Salt and the Dix River, which sometimes is part of the Salt, sometimes it's part of the Kentucky, and lots of exchanges. So the Kentucky River sort of stealing from the Cumberland, the Kentucky and Salt are sort of stealing back and forth from each other. And we can see this in the distribution of fishes. And so that got me thinking about this distribution of juvenilis. We might have a possible route of combination of vicariants. That's a change in the geographic landscape, would be stream piracy as an example of vicariants. So it looks like on the tree, there's this route that this lineage has gone to until they essentially kind of bumped up to the glaciers, and you have the additional species of Jeffersonii and Sloanii sort of as glacial relics, sort of little pockets of speciation. Then our other clade, call this the Lower Ohio clade. This is the one that the new species is resolved in. These all are confined to this area of the Lower Ohio River and Wabash system. With the exception of, there's one that has dispersed out of this. Faxonius propinquus has expanded much further beyond this after the recession of glaciers, but many of them have really small ranges within this area. And so there's sort of a shared unique history here. Our new species is sort of the most eastern representative of this, right below the falls of the Ohio. So there it is. But there's some weird things going on. It is actually genetically identical to two other species in this clade, Faxonius illinoisensis and Faxonius standardi from southern Indiana and southern Illinois. They are all morphologically distinct. Their gonopods do not look like each other. So I'm not necessarily saying they are the same species. I've never seen these animals. I wouldn't say that they're the same, but that is a possibility that we have to consider. But I think what's more likely is that this is an example of incomplete lineage sorting. Basically, these are really recently diverged because of the more recent formation of this habitat and the formation of the Ohio River. So I don't think they're necessarily the same species. We have morphological data to support that they are different, but I think this is an example of incomplete lineage sorting. Basically, there hasn't been enough time for mutations to build up that they are genetically different at this particular mitochondrial marker. Now, we could address this with population genetic data and say, are they separate? We'll work on a grant proposal to get the money to do that. There are other examples of incomplete lineage sorting. This is Faxonius christovarius, and this is Faxonius sandborni. Their gonopods look really different. One's really short and one's really long. And so interestingly, the ranges of these would have been highly influenced by this glacial lake type. So christovarius is mostly down here in the Appalachians or across the Appalachians, where sandborni up here on this side. I've talked to Günther, apparently they seem to be sympatric in one place, Tygert's Creek, which would have been part of this lake type. Genetically, they are indistinguishable. We only have two sandborni in here, but a whole lot of christovarius. But right now, they're inseparable at this particular marker. And so this could be another example of sort of rapid morphological change in response to this habitat change. As lake type drained, we now have this sort of low gradient streams that sandborni is more found in, and then the high gradient streams that christovarius is found in. Some of this data might actually show some cryptic invasions. This is a series of papers that were published in 2015, potentially introduced species in North Carolina and Tennessee. And unfortunately, some of them come out identical to this new species in Louisville. They're calling it Faxonius ronaldi because they didn't really know what it was. There's some conflict between the genetic data and the morphological data. But that was the name they settled on. We sampled ronaldi a bit more. It doesn't seem to be that. It's really divergent from the type locality. But the issue with the paper that identified these as ronaldi was that they did not actually include these other samples of illinoisensis and standardi in their data analysis. So they did not use all the available data. Those sequences have been available since 2006. So they sort of cherry picked, and part of it was the reliance on the subgenera. So they only did their genetic analysis and compared it to other things in that subgenus. Well, we know those subgenera don't actually work out on the tree. So their incorrect conclusion about what this is was based on exclusion of some available data. I don't know what it is, but it is genetically identical to this new species. It is morphologically different. It doesn't look anything like this new species. There's other examples of this with juvenilis, also found in potentially non-native areas in North Carolina. Some of these I'm not so sure actually are non-native. Some of them look like they could be, but some of them might actually just be range expansions. I don't know, I'm not sure. But really to get to identifying invasive species, you really need good reference databases and good phylogeography. This is one example where they were able to identify this non-native salamander that popped up in the Ozarks. And the only way they were able to do that is because they had really good comprehensive reference databases to compare it to. They were actually able to get it down to the county level. So they were able to, and they actually found some bait shops that sell this thing as bait. So we're not quite there with crayfish. We have like single representatives for species, but we really need to sample more across their drainages so that we can identify these invaders. There is extensive polyphyly in the tree as well. So we get the same species popping up in two different places. This is another example. The rusty crayfish pops up in 2 different places. These possibly could be misidentifications, but I think more likely they are cryptic species because the biogeography sort of makes sense with where these things were collected. So these rusticus and putnami fall out in that lower Ohio river clade, and they were collected in the lower Ohio River. These other ones fall outside of that. And so I think they actually are different species because they sort of fit with the biogeography. What this could indicate is that there's been convergence on the morphology. Since these are identified by the gonopods, there is likely extensive morphological convergence. So some of the key takeaways, Louisville is home to 2 endemic crayfish. It seems like it's, we're gonna call it a new species based on the morphology, although there is some questions about the genetic data. But the crayfish mitochondrial DNA does show distinctive phylogeographical patterns, and they might be better than any other group of organisms, better than fish, better than mussels. There are a whole lot more patterns that I wasn't able to talk about. I need like 5 hours essentially to go through all of them. These issues of incomplete lineage sorting and paraphyly might be common in crayfish mitochondrial DNA, and that polyphyly might be indication of cryptic species. And we need to take caution about just sort of using a blanket DNA barcoding. Just take a gene, what is it compared to, call it that. I don't think we're really ready to do that with crayfish until we put the legwork in and really completing the reference databases. It's also possible that the reproductive morphology of crayfish might change in response to environmental or habitat changes, and it might change faster than the mitochondrial DNA, which is why we might see examples of incomplete lineage sorting. So mitochondrially, they look identical, but they have really different gonopod shapes. So the gonopod might be more plastic and change faster in response to environmental change than the DNA mutation rate. And so this whole stuff, this entire hour of me rambling about this came from natural history surveys. They went out and surveyed for this, and basically boots on the ground and a keen eye. And there is definitely a lot more out there to discover. Also note, I generated almost no novel data. I don't know if you caught that, but the Jeffersoni, there was already 1 sequence of that available on GenBank. I generated about 29 more of them, identical. And our new species sequence was identical to several other things that were in that original 2006 study. So I generated no novel data. So let's just give you graduate students some confidence. It doesn't necessarily have to be novel. So some future work, I want to extend sampling, obviously increase those reference databases. Faxonius rusticus is one I'm really interested in because it is highly invasive in many parts of the country. It is native to here in Kentucky, but as you saw, it's polyphyletic, and it might be multiple species. And so Günther also tells me the colors are different. Some populations have the rust spot on the side of the carapace, others do not. So the color and the biogeography might indicate this is actually a complex of different things. And I also want to start targeting these stream capture areas, sample crayfish from this drainage and this drainage, across those paleo drainage divides, and look at the genetic differences across those. The Ohio River, if somebody proposed, I want to study the biogeography of the falls of the Ohio, I would have said that's dumb because nobody has ever found any genetic break related to the falls of the Ohio. And again, it's not really the bound, it's sort of incidental or coincidental. It's not really a boundary to the crayfish. But now I want to figure out other places like that to study the breaks between. And we really need some population genetics or population genomics for some of these groups, particularly like this new species, to see if they're actually reproductively isolated from one another. So there are a ton of other cool things in this tuft here and other cool things in the bottom there. There are Ozark species and other evidence of the Taze River. I was able to just talk about these 2 little tufts here. And so sometimes going through this, I feel like, I'm conspiracy theory, connecting dots and making connections here. And I just want to end with Arnold Ortmann here. Günther mentioned him in his talk. In my opinion, one of the greatest aquatic biologists that ever lived. He was a German immigrant, worked at the Carnegie Museum, really prolific in bivalves, crayfish, snails. And he wrote this paper in 1913, The Alleghenian Divide and Its Influence Upon the Freshwater Fauna. I found this one paragraph in here. So I'll just sort of summarize the naiad fauna. Naiads are the bivalves, the clams of the upper Ohio. Their patterns look like the modern day course. And that sort of makes sense because they use fish as hosts. So the fish also kind of look like that. So their patterns, the patterns from the bivalves are post-glacial. So after the glaciers receded, that's the patterns that we see. The crayfish fauna, however, indicates pre-glacial conditions. Their relationships are reflected by what the rivers were like before the glaciers were there. And then lastly, the Pleuriseridae, these are large river snails, basically are a mess. He says the natural affinities of this group are too obscure to permit any final conclusion. This is still true today. I basically gave up doing molecular work on snails because the mitochondrial work, the DNA comes out crazy. It makes no sense geographically. I can find two snails in the same rock and they'll come off different parts of the tree. So basically Ortmann here in 1913 summarized 15 years of my work in genetics and thousands of dollars in grant money. He already knew all the stuff that we're able to show with genetics. So another key takeaway, I generated almost no novel data here. But with that, I will take any questions.
Thank you, David. Great presentation. Really, really informative and quite fascinating. Folks, if you have questions, why don't you just call out, and I think that will also unmute yourself when you want to ask a question, if you would.
I have a question, David.
Shoot, Roger.